Do you want me to go ahead and start? Oh, okay, great. No. Some of you know I'm Mike Lynch by now, I hope. And this is my research talk. It's about a group of arthropods. I'm pretty sure a few of you know what an arthropod is. This isn't the same one that most of you work on. This is an aquatic microcrustacean. We work mainly on Daphnia pulex. I'll just sort of start this off with a...
You might drop the mic a little bit.
Drop the mic. How about this? Okay?
Can you hear back there? No, we can't. Never mind.
All right. How about this? Okay, all right. So first, just a little background on the system. We've been working on, if I get to the final slide of my talk, you'll see that I've been working on Daphnia for a long time. Early on was for purely ecological, limnological reasons, but now we're pretty heavily involved in population quantitative genetics and population genomics. And so we've had this big project going on that we now just call the Daphnia pulex Genomes Project. One of the goals is to sequence, we like this number, 96 genomes from 50 populations over the full geographic range of the species, which I'll demonstrate to you in a few minutes, and a few of its close relatives. And as we'll see, we have one population that we're using as sort of a litmus test to try and understand what happens over time in an undisturbed population genomically across the genome. And we're expanding this now to several other species to try and get a general feeling of how general our results are for pulex. This is what the bug is. So they're transparent. They live in the open waters of essentially every lake and pond throughout the world, as long as the salinity isn't too high. And I'll go through the life cycle in a second. Just for orientation, that's its head. That's the eye spot. And these are swimming antennules. That's how they get around the water. And they're filter feeders. So they filter all day long, 24 hours a day, in the water column, any kind of bacteria or algae that comes in. And some of the tools that we've been trying to develop is we're trying to get a list of the functional genes across the genome in quite a few species. We've got good reference genomes, almost telomere to telomere for about 20 different species now. We're doing a lot of work, as we'll see, on tissue-specific and developmental stage-specific expression of genes and promoters. We're trying to ascertain it turns out that ribosomes and mitochondria are central to a lot of unique evolutionary aspects of these species. So we're now trying to figure out the structure of the cytosolic and mitochondrial ribosome, mitochondrial proteome, and then we're trying to figure out the nuclear cyto interactions between the genes that make the products that produce the electron transport chain and so on. And we're trying to develop, like everybody else, we're trying to develop efficient tools for CRISPR-Cas9 so we can easily transform the lines from one type to another. It's not real easy because we have to inject the eggs of these bugs, and the eggs they're soft for about 10 minutes when they come out, and then they harden and poof, you lose everything. So they're reproducing every couple of days they molt. So every couple of days, these babies are developing in the brood pouch of the mom, and then they come out and she immediately puts in another brood. And then one of the big advantages of Daphnia is this one, sort of the mirror image of worms and flies, where there's not a whole lot known about the natural ecology. There's a ton of information on the natural ecology of Daphnia. Limnologists have been working on Daphnia for like over a century and a half. There's just thousands and thousands of papers. It's a big victim of aquatic ecotoxicological studies as well. But what we're missing that you other guys have are a good toolbox for molecular and cell biology. So that's what we're really putting a lot of effort into now. So that eventually we'll have tools for both molecular work as well as being able to do these kinds of studies in the ecological context. So just a little background. They've been around for quite a while. Daphnia have been around since about the Permian at least, and they're members of the order that we call Branchiopoda, which has 4 different suborders. So these are the tadpole shrimp. Some of you have probably seen these in temporary ponds. These are the fairy shrimp. Some of you have probably heard of Artemia. These are the clam shrimp, and then Daphne are members of the Schlötterer. This is just an example of the morphological diversity that you can see. Many of them have phenotypic plasticity, so they can grow huge helmets in the presence of fish. It's just phenotypically induced without a genetic change. This is one that's invaded a lot of lakes, including all the lakes, as far as I can tell, all the artificial lakes in the state of Arizona, called Daphnia lumholtzi. It's got this big spine coming off the top of its head, 2 strong spines coming out the side of its head. So a lot of interesting morphological diversity. The life cycle goes like this. Some of you might be familiar with aphids they're called cyclical parthenogens, and that's what most of the Daphnia I'll be talking about today. I'll give you one nice side excursion where this is not exactly the case, but for the most part, you've got a mom, and every few days she's producing daughters that are genetically identical to herself, and they grow up, and in certain environments, if you stress them or crowd them, the moms start producing boys. The boys are genetically identical to the mother, they've just gone down a different developmental cascade. And then the females can start producing haploid resting eggs, and then the boys get excited and they start fertilizing sexual females. And those resting eggs are called ephipia, and they're released, and they're desiccation resistant. They can last for hundreds of thousands of years in the environment. And that then completes the life cycle. So that's a sexual phase of the life cycle. And they're diploid throughout this whole phase, and that's just a couple of Daphnia getting it on to. He's picked the wrong female. She's not about to produce resting eggs, but nevertheless, like our males, they make mistakes. So here's a global map of the distribution of Daphnia pulex. So it's found throughout the northern hemisphere. There's very closely related species throughout Europe and Asia. They're also found in South America. So these guys are really globally distributed. These are the kind of environments you can find them everywhere, from a pond that has a dead cow floating in to a dune pond, to a saline pond near the shore, to these deep, pristine lakes. So they really live in a diversity of ecological settings, opening up opportunities to perhaps study the genetic basis of ecological adaptation. They've also somehow found their way around the world, probably in the last 30 years. So there's Daphnia pulex now in the Rift Lakes in Africa. They've worked their way into some lakes in China here. And New Zealand, we spent, a couple of years ago, I spent about a month sampling all over New Zealand. Daphnia pulex has invaded New Zealand, and it's taking over every lake in New Zealand, driving the 2 native species of Daphnia in New Zealand extinct. And it turns out it's just 2 clones, obligately asexual clones, taking over both islands. So that's turning out to be a pretty interesting thing. They were probably dropped into the lakes there by rich fishermen from North America who dropped in trout into these big lakes from California, which probably had some Daphnia in the water and seeded the lakes. This is related to these resting eggs I just mentioned. This is something you can do with this system. There's a few other systems you can do it with. These, as I mentioned, these resting eggs can survive up to 200 years at the bottoms of lakes. And some lakes that have high productivity in calcium-rich areas produce these sediments. These are called varve sediments, and you can just peel off the layers of a core from your lake, just like rings in a tree. And then in each layer, you can sift out these kinds of eggs, and you can grow them up in the lab, and you can literally have a couple hundred years of evolution alive side by side in the lab, maintained clonally forevermore. And you can get DNA out of eggs far beyond that. So that's sort of a background of the system. Any questions about the basic biology of the bugs? Okay, so this is sort of the roadmap where I'd like to be going. I'll first just give you some general insights, I hope, into the population genomic features we now know about with these bugs. Then I'm going to take a little excursion on a new discovery we've recently made. It turns out there are some Daphnia. Surprisingly, this was just discovered after like 200 years of research. We've discovered some genotypes of Daphnia out there who are unable to make boys. So they can, this is like gynodioece in plants. We have genotypes that only have female function and others that have male and female function. And I'm going to talk about a long-term population genomic study we've been doing. We've been studying the changes in allele frequency in one population for a decade now, and that's yielding some pretty interesting results. You may not think they're interesting, and I'll turn my back when I'm done presenting these data, and Bruce will take a tally so you can tell me whether we should keep going with this study for a while. And then this last part was something I never really fully got to my last talk. It's related to this release of hidden genetic variants and genetic slippage of mean phenotypes in response to sex. So it relates us back to quantitative genetics. We'll see if I get there. So first of all, the basic population genomic features of the system. So this is from one study we just published a couple of years ago. What we typically do when we want to study a population, we go out and collect about 100 clones or so, grow them up for a few weeks, and then extract DNA. We barcode them, and then we just multiplex sequence the whole population. And so we are literally getting sequences of about 100 individual clones from each population. This is a study of 10 populations of Daphnia pulex. This is the Midwest of the United States. So these are the Great Lakes, and these are just south of the Great Lakes. And what you can see here is there's population subdivision and there's isolation by distance. So I've color-coded these populations over here. This is sort of a crude tree based on whole genome sequencing, whole population genomic sequencing and allele frequencies. And you can see that the lakes or ponds that are fairly close together come out fairly similar positions on the tree. So pretty good concordance between the tree and the geographic distances. You can look at FST. So this is sort of isolation, slow isolation by distance. This is the average pairwise FST between these populations with geographic distance. And using this information, you have to be careful with FST. People have been talking about this, and I'll get back to this in a second. But one problem with FST is it's biased unless your allele frequencies are fairly high. And so you'll see we're only using FST measures with allele frequencies above a certain value because there's an upper limit, it turns out, to FST in a study like this. And you want to get away from that upper bound problem. This is a plot of the number of private alleles in each of these populations as a function of the minor allele frequency for the total population. So by that, what we mean is we're looking at a metapopulation frequency of, say, 0.02, and given that in each of these populations, how many populations have a private allele in that window? Does that make sense? A private allele means the allele is restricted to an individual population. It doesn't mean it's a singleton, but it's restricted to just one of the 10 populations. And then you can plot the number of private alleles against the minor allele frequency in the total population. This might seem nonsensical. It turns out extremely useful because Monty Slatkin published this nice paper, about 30, 40 years ago, showing that with this kind of information, these kinds of regressions, you can estimate the population migration rate under an island model, which is what we have. And using either approach, this is the standard formula I think some of you have seen for an island model. This is what FST should be for neutral markers. And so that's 4 times the effective size times the migration rate per population. And so we've been able to estimate M 2 quite different ways. And both ways you get a migration rate of about 80% of an individual per population per year. So there is migration going on. It's pretty low level. So the fractional migration rate, if you divide that by the total effective size, is about 10 to the minus 6. That's what little m would be, about 10 to the minus 6 per year. And what that tells us, Sam was kind of alluding to this. If you think about the power of the power of migration, that's sort of a measure of how to the extent to which migration can overwhelm local adaptation. And so in our case, with that level of migration, efficient selection is going to require, it's only going to require selection coefficients greater than 10 to the minus 6. Does that make sense? You want to have the strength of selection operating a nucleotide site to be higher than the rate at which migration is swamping out the effects of selection. Okay, so that's the basic population structure. These populations are, I should say that when we sample these populations, in fact, I should back up a second. All these populations that I'm talking about are temporary pond populations. So they fill up in the spring each year, and then they dry up, usually by June or so. And so they're forced into a sexual phase of reproduction every year, because it's the only way to produce the resting eggs as we get around to the next year. So these are like annual plant populations almost. The pond fills up, there's differential clonal expansion, resting egg production, random mating, and it all starts over the next year. And these populations are very, very close to Hardy-Weinberg expectations when we collect them. We collect them very early in the season, the very first things to emerge from the egg bank in the population. And when we do, they're very, very close to Hardy-Weinberg expectations. So that's FIS, this thing we call the inbreeding coefficient. It's a tiny bit lower here, but even here it's only minus 0.01. So that's a very small deviation. And this is probably simply due to the statistical bias that I talked about. And here's the measures of FST. And you can see for rare alleles, which is often most of the alleles in your population, these measures of FST are downwardly biased. These are the theoretical expectations. And it has this weird behavior that's been pointed out by a number of people. But once you get into a certain threshold allele frequency, everything falls in line. And so that's generally where we try to reside when we're using FST for various applications. There's a tiny, if you can see the blue dots here, those are silent sites, and that's the lowest, but there's very little difference between FST for silent sites, amino acid replacement sites, and so on. So studies like this, you've seen this before, you can go into your populations and measure standing nucleotide variation, and we break it up into sites in intergenic DNA, deep intergenic DNA, silent sites in protein coding regions, replacement sites, certain sites in introns. We've done a lot of work. There's certain regions in introns we're pretty sure are evolving in a nearly neutral fashion. You can compare that with the silent sites. And then in the 5' and 3' UTRs. So the UTRs are the regions of a gene that are transcribed, but they're right adjacent to the coding region, but they're not translated. And so they're under a bit more constraint. We know that not surprisingly, we've done mutation accumulation experiments on Daphnia, so we know the mutation rate for nucleotide site. And then if we set this equal to 4 NEU and divide by the mutation rate, a rough average estimate of the effective population size for these bugs is about a little over 500,000 individuals. And you can see it's fairly constant across the whole metapopulation. And I already showed you this before the last class I taught. These are these historical measures of effective population size in 8 of the populations. And they're quite flat going into the distant past. But if you really home in on these things, this is on a log scale now, it looks like there was a slight dip and then somewhat of an increase, perhaps in the last, I thought I put the numbers up here. All this happened perhaps in the last 10,000 generations. There's about 4 generations with these Daphnia populations per year. So this all happened maybe in the last 2,000 years or so. Otherwise, things in the deep past look pretty stable. We've now got pretty good genetic maps. These are crossover maps that many of you know how to make. I just want to show you that we have them. They're quite linear, except in these red regions there, those are the centromeric regions. So at least from the standpoint of crossovers, the centromeric regions have very little recombination going on. But from population genomic data, we know it's it's by no means zero in those regions. I want to mention something about linkage disequilibrium, and in doing so, I want to sort of advertise a nice, powerful method for measuring linkage disequilibrium I think most of you aren't aware of. You can measure linkage disequilibrium. You're used to hearing about people measuring LD based on population samples. And you've got everybody's genotypes at 2 loci, and then you look for correlations between haplotype frequencies and so on. And that's worked quite well. It's pretty noisy, though. And it requires a lot of assumptions. You can measure linkage disequilibrium really well for a single randomating population, just with a single individual. And the way you do this is you just look at your individual and you go up and down the genotype, and every site, it's a diploid, is either a homozygote 0 or a heterozygote H. And now you've got the geographic distribution, so to speak, of heterozygous sites across the genome. And then you can measure this thing we call the correlation of heterozygosity, or correlation of zygosity. And it has simple explanation. So if everything were Hardy-Weinberg, or in linkage disequilibrium, if pi is the heterozygosity, average heterozygosity, then the frequency of double heterozygotes at a certain distance is just the probability of being a homozygote squared, and pi squared would be the probability of a double heterozygote, and that would be the probability of one site being heterozygous and the other homozygous. And what we do this for all sites that are adjacent, make our calculation. These added terms here, this is due to the correlation of zygosity. This will, so some of you are familiar with FIS, this will look familiar to you as the FIS sorts of equations, which is, FIS, remember, is a correlation between gametes at single sites. So we can, these are the expected values of double homozygotes, heterozygotes, and mixed pairs. And what you do is you simply go for all sites, all pairs of sites that are adjacent, and make these calculations using a maximum likelihood method that we've developed. All sites, all pairs 2 sites apart, 3 sites apart, 4 sites apart. You just do this over the whole genome, and you get a nice, beautiful profile built up. And the reason this is interesting, you don't need to phase your genotypes. There's no need to phase. You're either a heterozygote or a homozygote. You can apply it to single individuals. Yep. Oh yeah, it can be used to infer that, actually. But it's not, the method isn't limited by population size. Okay? You can apply it to any diploid species. And in fact, I'll show you in a second how you might work back to get the effective population size issue. There's worries in a lot of these other techniques that try to measure LD because they make inferences about missing sites, and there's typically missing sites even in the refined days of genomics we are at, and there's no problem with that. You just skip over those sites here. And the theory has been worked out for the meaning of these coefficients in terms of population size, recombination rate, and mutation rate, which I'll show you in just a second. And Bernard Halbelt has written a very nice program that you can just use. You just plug in for every site in your genome the number of A, C, T's, and G's seen at each site, the number of reads, it factors out the error rate and everything. It's very, very nice, easy to use, and quite powerful. Anybody know Curtis, remember Curtis Strobeck? Bruce? Christian, okay. He's I don't know, he's probably not as old as me, but he's an ancient population geneticist from North America who's done some really nice stuff. He wrote this paper back in 1978. It's effectively the expected value for the correlation of zygosity before anybody ever imagined we would actually have these data. And it turns out it's a function of theta for NEU. It's a polynomial in the numerator and the denominator, and it's a function of rho for NEC. So if you know U like we do in NE, or if you just know theta, then you can estimate rho from your correlation of zygosity. And that C is going to be the amount of recombination per site times the distance between your sites. And so we've measured LD in a number of ways in our Daphnia populations. This is the average R squared value. These are all plotted as distance between sites. And this is a problem with R squared measures of linkage disequilibrium. They're functions of the allele frequencies being used in the analyses. I think Bruce was sort of alluding to this. Unless your allele frequencies at both sites are identical, you can't get a correlation of one. It works out. And the more the disparity, the bigger the problem. So we do this in different windows, minor allele frequencies between 0.4, 0.5, 0.3, and you get these nice curves. We have a lot of data here. This is based on 10 populations. These are the correlations of zygosity. They behave really, really nicely. And then as we'll see, we have a population that we've been studying for many years, and we can measure LD in a different way. We look at the correlation of allele frequency change between two sites in 2 years. And then if they're linked, you would expect the behavior of this allele and this allele to behave in a coordinated fashion. All these things drop off with physical distance until you get out to about 10 to the fifth, and then you're just sort of bottomed out. So about 100 kb is sort of the limits of meaningful LD in these populations. One thing I want to emphasize here that there's not a lot of Daphnia population geneticists out there. So usually when our paper goes out, I'm pretty sure they're Drosophila population geneticists reviewing the paper. And we always get this criticism, well, these bugs are nuts. Nothing can be general about these. And what about the recombination rate? So you can do a back of the envelope calculation and easily show that the recombination rate, the mean recombination rate, averaged even over the asexual generations in Daphnia is about 30% higher than it is in Drosophila melanogaster. There's two reasons for this. One is half the individuals in a fly population have 0 recombination rates in each generation. And flies have what, 3.5? I don't know how you want to count it. How many chromosomes do flies have? It's a small number. Daphnia have 12. So there's more recombination going on per chromosome because of this one crossover per arm. So let's just say they have a comparable recombination rate per nucleotide site for an arthropod in general. And then you can, with lots of data like we have, you can start looking for fine scale linkage disequilibrium using population genomic data. And there's programs like ML-HAT and ML-RO and so on. And I think most of you've seen these profiles before. This is just along one part of one chromosome. Chromosomes are about, I guess the average the genome size is about 160 megabases. Divide that by 12, so I guess the average chromosome's something around 13 megabases. This is part of one chromosome. And these are measures of the population recombination rate, rho. Yeah.
Sorry, I just had a question. You probably said and I missed it, but the NE and the recombination rate, are they per generation or per sexual generation?
I think these would be per sexual generation. Yeah.
Do you know what that is? I mean, or is it?
Do
you know what that is? Is it sexual generation once per year or something like that?
There's one sexual generation per year.
Okay.
They're like annual plants, right? They hatch out of the sediments each year. They've been dried out for what, 6-8 months, and they hatch out. Clones expand a bit, and then they all go sexual, and the pond dries up. And anybody who didn't produce resting eggs is a goner, basically, at that point. And so you've all seen these plots, I think, probably. These are based on LD, and these are measured. There's 10 plots here for the 10 populations. And so this is a noisy measure. I mean, you'll be seeing many examples, and I'm not going to apologize for how noisy some of our data are. This is just the nature of needing very large sample sizes to get good estimates of population genomic parameters. But you get all these peaks and so on, but this is an average from this plot, and you can see the peaks come down a bit, and there aren't huge differences between these numbers. I've really magnified things. And in fact, that's the general point here, is that there's very little evidence for, some of you have heard about recombination hotspots, say in mammalian genomes and so on, and we don't have them. There's hardly any in flies either. So the recombinational landscape is quite flat. And just to give you an idea, this is the cumulative, if just going from the sites or the windows with the lowest recombination, just adding up to the whole recombination map, that's how the plot goes. And that's pretty much how it should go if everything's random. And this is what you get for humans, because there's a very small number of hotspots that account for most of the recombination. And it gives you that deep bend in the curve. And you get that for mouse, and that's what you get for flies. So there's a little bit more of a hot spottiness going on in flies than Daphnia, but not much. And then you can, if you want to, you can make conservative estimates of what you want to call a hot spot in Daphnia, but at best you would call these lukewarm spots, because this is the frequency distribution of the recombination rate in these spots compared to the average. And you can see this is only 6x normal above the average. So this could just be noise. So it's a pretty homogeneous recombinational landscape. Okay, so that's background in population genomics. Any questions on things up to now? I'm just trying to give you a little background on the system before I get into two special studies. Okay. So as I mentioned, I gave you the Daphnia life cycle, the typical textbook Daphnia life cycle a few minutes ago, but we've recently discovered a novel mating system in Daphnia, so I have to expand a bit. So this is the life cycle I just mentioned to you, cyclical parthenogenesis. Most of the year the population's reproducing clonally, and then a phase of sex, and then close the loop, and you do this the next year. What I won't talk about today, but we've worked on quite a bit, some of these Daphnia pulex have given up the ability to have sex at all. They're obligate asexuals. So instead of this mom, when she produces a resting egg, instead of it being a haploid egg that needs to be fertilized, it's a diploid egg, genetically identical to her, and it just closes the loop. So they're obligate clones, ameirotic parthenogenic clones. Some of them still produce males, which is quite interesting because these moms clearly have some kind of meiosis suppressor. And they can produce sons though, there's just a different pathway that turns on male production. Those sons often produce haploid sperm. They can then transmit the meiosis suppressor by backcrossing into a sexual species. And this gives rise to what we call contagious asexuality, because it's a dominant sex-limited meiosis suppressor. So half the kids that it produces when it mates with one of these will turn out to be obligately asexual. And for some reason, all of Canada has been turned, all Daphnia pulex in Canada have been turned to obligate asexuals, with a few exceptions.
How long did it take?
That's a good question. Let me know if you can think of a way we could do that. I think you could probably do it with mitochondrial phylogenies, or we haven't looked into that. Longer than my lifetime, I'm pretty sure. Okay, this is what I want to talk about, this thing here. We recently discovered that some of our Daphnia are unable to produce boys. So they still produce, when crowded, they'll still produce females with haploid eggs, but they're reliant on males produced by other clones in the population to fertilize. So this is, as we'll see, it's a dominant gene that prevents male production. And I like to think of this as being comparable to the mating system in plants called gynodiece. So in plants, there's a mating system where some individual plants make pollen and ovules, and other individuals just make ovules, right? That's called gynodiece. What we have are some clones that only make eggs, haploid eggs, and others that can go through male and female function. So it's sort of an analog to gynodiece in plants. And what we're trying to do now is to figure out what caused this genetic change. It's quite remarkable that people have been studying Daphnia for so long and no one had actually realized this. I mean, it's hard to prove a negative, of course, but we have some good juice now that I'll mention in a second. If we squirt it in the water, they're going to get super excited, and they're going to, no matter how much they want to resist, they're going to produce boys. These guys don't do that. And so, again, here's the mating system. So a lot of individuals are doing both of this, but some can't do this. And this is the model we're working at. Turns out we think a very simple model, as you'll see, although we haven't figured out exactly the molecular mechanism. But what we're saying is the male producers, the normal Daphne, we'll call little m, and the non-male producers are big M, little m. And so you cannot have a surviving population of just big M, little m, if it has to produce resting eggs, because there'd be no males in the population, right? If it's a permanent lake, they could go almost to fixation if they don't have to produce resting eggs, but not in the kind of populations we're talking about. There's no big M individuals, right? So this is like a sort of like a sex chromosome, but as we'll see, it's not a whole chromosome. You can't have this because these can't mate with themselves, okay? And then there's variation in the frequency of this type. Some populations, like the one I'll talk about in a few minutes, don't have any of these. And then some others have frequencies as high as close to 50%. We don't know the temporal dynamics or anything yet. Something that probably merits studying. In fact, I think this would be a really good model system for trying to understand the evolution of mating systems, because we have such diversity of allele frequencies and it's operating at such a rapid time scale. Okay, what about the gene? So fortunately, as I've said, we've been sequencing a lot of genomes in this species, and fortunately, Daphnia can be kept in the lab in a clonal fashion indefinitely. And so a lot of our populations that we sequenced all the genomes 4, 5, 6 years ago, we still had alive as clones in the lab. And their genomes had already been sequenced. So this is the ultimate GWAS, right? You just take the different clones, find out if they produce males, look for markers that are unique to the non-male producers, as simple as that. And it turned out to be, this is about as good as it can ever get, I think, in GWAS. It turned out in the first population we did this study in, I think we had about 80 individuals, maybe about 40 were non-male producers and 40 male. And we found 1,600 NMP-specific markers. In other words, those were markers, alleles that were only in the non-male producers, never in the male producers. And then we had other populations. 4 other populations we had these polymorphisms in. So we thought, can we narrow it down and do the same study in 4 other populations and do the Venn diagram thing and see how many of these, how far we can narrow the 1,600 down, looking for markers that are non-male producing specific in every population. We got it down to then about 132 SNPs and 59 indels that were perfect markers in 4 different populations. And of these 132 SNPs, only 15 were located in coding regions. And 8 of those 15 cause amino acid substitutions. And all of those were in a single gene, gene 8960, because this is we knew it was a gene in there based on its open reading frame and expression, but we have no idea what this gene does, and we're trying to figure this out now. It's an important gene. I don't know what you mean by a homolog, but it seems to be a Daphnia-specific gene. So far, we don't find this gene outside of Daphnia. It's got interins, so, well, I'll give you a little more history, but it's deeper history we don't know.
So perfect knockout.
So, yeah, exactly. And this is one of our first applications of CRISPR-Cas9. And I'll show you why in a second. So gene 8960, that's the male essential locus. The M allele, I have 3 Chinese postdocs in my lab. So and we live in Phoenix. So they named this gene Feng, the big M allele Feng for the male phoenix. Yeah, as a candidate locus for now.
Yeah, but it could be also regulatory.
Yeah, it could be. But in fact, I'm going to talk about one more gene that answers your question maybe. But we just decided to start, we had to start somewhere because in this region, I think I've forgotten, there's about 80 different genes and we want to start somewhere. And it just seemed odd that this is a rapidly evolving gene. So that was the rationale, nothing too much deeper than that. Okay, so big M is called Feng now. We have a lot of these genome sequence, including a lot of the non-male producers. So we can make a genealogy now of the normal alleles, the little m alleles, and the big M allele. And it turns out that so these are clones from 10 different populations. And so these are the big M alleles. So we were able to make the haplotypes. Remember, everybody's a diploid. And so if you're a male producer, you've got 2 haplotypes here. And if you're a non-male producer, you've got 2 haplotypes as well. One is this big M allele, and the other is the little M allele. And even if you're a non-male producer, your little M allele comes out here in the clade with the little M allele and everybody else. That makes sense because when you cross each generation, you're just crossing into the little m individuals. But what's interesting here, the big M allele clade is monophyletic. Okay, so that was the first thing that jumped out. That's consistent with this allele having a lack of recombination. Otherwise, all these things would be shuffled together. So it seems to be a monophyletic clade, apparently lacking in much, if any, recombination. The M allele seems to be introgressed from a neighboring species, Daphnia pulex. It's an introgressed allele. Daphnia pulicaria. Daphnia pulicaria is the closest relative to Daphnia pulex. It lives in big permanent lakes where pulex lives in temporary ponds, but you can hybridize them in the lab. So somehow this allele got into these guys from pulicaria. There's at least one pulicaria in this clade so far. And because we know the mutation rate, we can date the divergence of these clades. And that event there is about 0.8 million years ago, is where the Daphnia derived allele separated from the pulicaria. We can also date the base of the existing clade. This is based sort of the coalescent of the big M allele, and that dates about 0.2 million years ago. So if you divide by about 4, that's the number of generations ago, how far this goes in the past. So that's the gene genealogy. Any questions to this point? Yeah, that's one of the big questions we need to figure out. Oh, one of the first things we're going to look is there some kind of meiotic drive going on?
It sounds like sun.
Yeah, we don't know the answer to that, but I don't have as many ecologists in my lab as I used to, so we know that's the exact question we got to figure out. Why is it so successful in some populations? Is there some kind of frequency dependence going on? Because you imagine this can't produce males, and if it finds in an environment where there's lots and lots of males, put it all into girls, right? So.
But do his offspring have more advantage because they,
who knows? Maybe there's some allele in that linkage group there.
I mean, it's an evolution, sex argument, right? Instead of crossing with yourself, you're crossing with someone.
Well, yeah, if you want to invoke avoiding inbreeding depression, this cannot inbreed. These other Daphnia, by the way, can inbreed. You can take a clone and get it to produce boys and resting eggs, and they'll self-fertilize. So this thing can't inbreed. There's a lot of potential reasons for it being there. One thing I'll just mention now because I don't want to forget. A researcher named Christoph Haag, that some of you know, several years ago, discovered a similar phenomenon in a European species called Daphnia magna. Hasn't been well worked out, the molecular mechanisms or anything, but interestingly, it looks like it's a completely different mechanism in Daphnia magna. And it's possible that these kinds of things are continually cropping up in populations if there's an ecological advantage occasionally. Okay, so I think I failed to mention, sorry, this is not behaving greatly. Yeah, I'm not even sure if I mentioned, all these markers, by the way, that I said we found, they're all in one region, the tip of one chromosome, chromosome 1. They're all linked together, so it's one big linked, or small linked, non-recombining region. And there it is again. We have really good genomes sequenced for multiple Daphnia pulex now, and they're all collinear. So that's the big M chromosome. And so you might think about that as, if you want to call this a sex determination region, I don't know, I think of it as sort of a mating system region, but everything's collinear here. There's no inversions or anything going on. And you can look at how much variation is in the little m allele relative to other alleles in the genome, and you might expect it to be 25% less, right? Because in typical matings, there's 3 of these, 1 of those, and there's 4 of these. And so there is, in fact, about 25% reduced heterozygosity in genes on the little m chromosome. And there's elevated heterozygosity in the big M allelic region. That may be because of gradual mutation accumulation in this region, because there's no recombination. It's going to pick up mutations, maybe even slightly deleterious by, maybe Muller's ratchet-like effects. Oops. Okay, I just said that. I've already mentioned reduced recombination in this particular region. This is the linkage disequilibrium profile as a function of distance between sites. And these lines up here are for the non-male producing region in different kinds of clones. And this is in this is in the recombining region of the chromosomes. This is in the non-recombining region where the big M allele resides. And you can see it's got elevated linkage disequilibrium, which is just consistent with the no crossing over going on in there. How did this all happen? Well, it turns out that most Daphnia species have 10 or 12 chromosomes, and there's a split between 2 subgenera. One of them is this popular species in Europe called Daphnia magna. It's got 10 chromosomes. Daphnia pulex and many other related species have 12, and this is due to a fission of a previous chromosome in the other subgenus. And as a consequence of that fission, so chromosome 2 over here has become 6 and 1 here. And as a consequence, the end of these chromosomes are the centromere. So that's where we think the centromere came from, on the end of the chromosome. Other Daphnia chromosomes are metacentric with the centromere in the middle. But this one's, what we call it, acrocentric, telocentric, something like that. Okay, so what's going on? We're trying to now get a molecular dissection of all this. And we know something about the neuroendocrine pathway for sex determination in arthropods. And I'm not going to go through the details, but it goes something like this. And we know the genes for these, and we've looked at a lot of aspects of the genes for these. And the important thing here is there's a thing called methylfarnesate produced in this pathway. And for most Daphnia, even if they don't want to produce boys, if you put them in the wrong photoperiod or whatever, you can buy methylfarnesate from Sigma. You don't want to get any on you, I guess, because what happens with Daphnia, they just start producing boys and only boys. So we use that as a quick test for ability to produce males. So it's a hormone. And what we find is that the big M allele is basically non-responsive to methylfarnesate. And so if you hit normal male-producing clones, that's where they are with no methylfarnesate, and then everything doubles up if you add methylfarnesate. And if the non-male producing clones, that would be the big M allele. There's hardly any response. So we think that the mechanism associated with non-male production is somehow associated with just expression of the big M allele. And we now have a lot of information on tissue-specific expression of genes in all kinds of tissues in the bugs. And these are all receptors and so on that I just pointed out. And there's gene 8960. This gene 8960 is one of the most highly expressed genes in the entire Daphnia genome. And it's unannotated, we don't know what it is. And what's interesting, it's really highly expressed in the swimming antennae, so up in here, and in the carapace, the shell that's around the bug. So it could have something to do with environmental sensing. There's a lot of neurons out here in these swimming appendages, and who knows what's going on in the carapace, but it's constantly being bathed by the water they're swimming through. So this is, I guess, the last slide. I've told you almost everything I can tell you about the system. We're really trying to dig it apart molecularly now. But we recently discovered another gene right next to Feng that one of my Chinese postdocs decided to name Huang for the female phoenix. They're the Phoenicians, I guess is what we might call them. And this one, so Feng knocks things down in males, and Huang is differentially expressed in females. It's hardly expressed at all in males. So, and it's got one of the highest evolutionary rates of any gene in Daphnia pulex. So it could be that this is sort of a modifier of the whole mating system. This thing stops you from producing males, and maybe this enhances female function. We don't know exactly what's going on yet, but that's all we know about this system, and we're sort of hoping to get funding now to further evaluate the methods and that the bugs go about going down this mating pathway. There could be real applications for this, say in biological control. If you could figure out a way to prevent bugs from making males, that would be the ball game for your pest. It would probably also select for parthenogenetic lineages, but anyhow, that's where we're going with this. Any questions at this point? Okay, now it's going to be a big switch. I'm trying to figure out what time it is. Okay. So we've embarked on this study in one particular population. I'll go through some of the data that we're looking at now. So everything I talked about to now is just one sample in 1 year from different populations. And I've been interested in knowing what goes on in a natural, undisturbed population if you just start thinking about the behavior allele frequencies across the board. I know you're all very interested in directional selection and posing specific forms of selection in the lab on fly populations and other populations, and that's been really informative. But it's surprising how little we know about allele frequency changes just in a normal, undisturbed population. And so what we're trying to do is to understand long-term temporal dynamics of allele frequency change across the whole genome. This relates back to, as we'll see, to a model that was suggested many, many years ago. I don't know if these guys thought this would ever turn out to be reality. Remember, this is before we could even do allozymes when this work was set out, but these guys worked out diffusion theory for a situation of what I think Dan Hartle first called quasi-neutrality. The idea is here there might be a nucleotide sites where the long-term average selection coefficient is 0, therefore we call it quasi-neutral, but there's meaningful fluctuations up and below 0 in any one generation. So there's significant temporal variation in the selection intensity. And in the models that have been developed so far, there's no covariance in selection across time intervals. And this can be worked out, of course, but I'm just saying that some of the things I've been talking about were actually envisioned many years ago in models by these guys, Carlin and Levickson, Jay Taylor, and a few other people. And what I want to emphasize here is we're going to be looking at the behavior of individual markers, SNPs up and down the genome. But remember, LD goes out to about, what did I say, 100 kb, which is going to contain a few dozen protein coding loci. So any of the selection that we are able to measure here, of course, is got to be due to any direct selection on that site, but selection on all the link sites as well. You can't separate those out. It's a standard problem in evolutionary genetics. This is the population. It's in a nature reserve in state of Indiana where it used to work, Portland Arch Nature Preserve. It's a very old, you can tell from the geology, it's a very old oxbow pond, and it typically just has water in it 2 to 3 months. Each spring, we go out every week or so early on because we want to catch the bugs as soon as they hatch out from the sediments before they've had babies and selection started to operate. And then we sequence 96 genomes. We've now sampled the population for 12 years. And the data I'll talk about today is only for the first 10 years of these kinds of studies. And most of the early, there have been super advances, of course, in the economics of genome sequencing. So most of our data are based on 10x coverage, but we're now sort of graduated about 30x coverage at less the price than when we started the study. So the idea is this. Just think of a single nucleotide site, and these are the allele frequencies in the population over some series of years. These are our sample sizes, and because the sample sizes are finite, we can only use maximum likelihood procedures to estimate what the true population frequencies are. So these are the data we have to work with, like anybody else, estimates of the population frequencies. And then we estimate the selection coefficients between these different points at each interval. So there's 10 years, so we can estimate 9 selection coefficients for any nucleotide site. We use this equation here. This is the discrete form of allele frequency change for an allele under additive effects. We use this because, in fact, these generations are discrete, right? Every year, population goes sexual. We wait till the next year, and so on. So we're looking at changes between the sexual generations. And if you rearrange this, you'll get this. But you have to be really careful with these kind of estimators, and that includes the logit transform that Sam pointed out before, because this estimator, the selection coefficient, it's got ratios in it. And when you've got ratios with sampling error, that will cause bias if you don't correct for the bias. And so we were able to work out what that bias correction term is. And so that gets plugged in as well. It's a function of the sample size. And we know from computer simulations that the estimators we're using are now unbiased. And so what we're going to be looking at is the sort of aggregate results of mean S estimates and temporal variances in S for about 1.5 million nucleotide sites. So there's about 10 to the 5th nucleotide sites per chromosome. These are the results for the mean values of S for all these 1.5 million SNPs, and I've divided them up into windows according to minor allele frequency. We aren't quite prepared to say we know the ancestral allele here, so we condition everything on the minor allele frequency. I think in another year or so, when we get more L groups, we'll be confident of what the ancestral and derived allele is. But here you see, these are these distributions. That's a mean value of 0. And you can see on average, these are close to 0, but they're not quite 0. So averaging over all the sites, the average minor allele is under positive selection. The minor allele is under positive selection. This is the average, of course, of about a value of about 0.003. There's slight skew to the right here. And these are meaningful values of selection. Remember, the size of the affected population tells us that the drift barrier is 10 to the minus 6. So that's less probably than the width of that line there. So this is real selection operating on these sites. And the range gets a little narrower. So what are we looking at? Down here, this is for pretty rare alleles. And then as we go to very common ones up here, you can see the distribution's getting somewhat narrower. I don't think that's due to anything biologically meaningful. As you get to higher allele frequencies, there's less sampling error in the estimates of S. So that's where the data are. So they're not far off, at least at face value, from potential quasi-neutrality scenario. Keep in mind that these selection coefficients are the net effects of selection in a region around each nucleotide site.
Mike, do you think the sampling error also has the asymmetry? If the asymmetry seems to be large, it's probably the lowest frequency.
I'd have to check that. Maybe not. I'd have to go back and check that. I'm not sure if the sampling variance is symmetrical. Thank you for asking. I'll go back and check that. I think you can see the tail's a little bit longer over here than it is over there. Now, we also want to estimate the variance in the selection coefficient. So for every site, we have 9 estimates. And we can calculate the raw variance of those 9 estimates. And those are, a variance is a positive number. But you have to be careful here because a lot of that variance among those 9 estimates is due to sampling error in the estimates. And you have to subtract that from the raw variance. And this is what's called a method of moments estimator. And when you do this, you do get a lot of negative estimates. You expect that because if nothing was going on, they should be centered around 0. Okay, so you get a lot of negative estimates, but when you take the averages, they come out to be on average positive. And this is just a summary for all the minor allele frequency classes of the behavior of these things I've been talking about. So these were the means that I just talked about. And you can see they're all somewhere between 10 to the minus 2, 10 to the minus 3. And these are the variances, temporal variances. These are the averages over all sites of the temporal variances. Sorry, this is a mouthful. Is people following what I'm saying? Every site we have an estimate of average S and variance of S over time. And this is the average of those over all sites. And they're hovering around 10 to the minus 2 for the variance of S. So it's pretty big. So we have an S in year 1, an S in year 2, an S in year 3, an S in year 4. And we take the variance among those. We're looking for between year variance in the strength of selection.
So from T to T plus 1?
T to T, yeah, T0 to T1, T1 to T2, T2 to T.
Average over all those time series that you
Yeah, for each site.
Excuse me?
have. Do you know correlations between years?
That's a problem you have to be careful about because if you use the data from T0 and T1, you don't want to use T1 and T2 because they're using the same value. So we split things up actually so there's no overlap in any one estimate.
Your figure showed different. Beforehand, when you were showing with that, you do the 10 estimates.
Yeah, well, we do the estimates, but in calculating the variance, we would look at that, we would skip over to always. And so there's no interdependence. Correct. Right. I have to go back on that. We might have even skipped over for S. What we'll typically do is we'll do the odd ones together, and then the even ones together and average over that. So neither one has got this interdependence, but there's this problem with strings of data like this if you're linking things together.
could you look at the synchronicity among these different locations, like if they're correlated among locations, selection coefficients?
Location, you mean among times?
You have different, oh, it's just one population.
It's one population.
Because, can you show the next graph, the next slide?
Oh, by the way, the reason for this is I'm going to have you take a vote later on. Our plan is to keep this study going as long as we can, but I don't live in Indiana any longer. But I have a colleague back there who's still collecting. And what I want to know is, do you think it's worth keeping the study going for another 10, 20 years? Just be thinking about that. That one?
What's the blue? No, sorry, the next one. The blue and the red then?
Yeah. Oh, this is a different measure. So this is the variance in S among sites. The variance falls out fairly similar. So right now, today I'm just talking about this and this. Okay? And what I want to estimate in terms of the noise here, just to give you an idea, only 8% of the raw, you get these raw sampling variances of S's. And then you have to subtract out all the variance just due to sampling error. And only about 8% is left. Okay? It's very hard to estimate a variance, right? Most of you know that it's hard enough to estimate a mean. You need a big sample size, but you need even a bigger sample size to estimate a variance. So these data will get better and better and better as we add more and more years, but this is where we are. We've tried to be really careful in developing unbiased estimators and so on. Okay, this just relates back to what I talked about with LD before. We've got average S's at each site, 1.5 million sites, and then we can say how is the S value that we've calculated at one site correlated with a site 10 bases apart, 20, 30, 40, 50. This would be due to LD, right? And you can see that the correlation drops off and it's essentially zero after about 100 kb, which is where I suggested before the sort of the vague length of an LD track is. Okay? All right, so out here, well, if 100 kb track mattered, if you're one site, there might be something like 5 protein coding genes on each side of you, just to give you an idea how much territory is in there. Now, another question that one can ask is, I just talked about the temporal variance in S. And then you can ask, well, what about the temporal covariance? So what about in adjacent years? Does S tend to be similar? Is it less similar, 2 years apart, 3 years apart, 4 years apart? There's some kind of predictable pattern. Well, there's a ton of data here, so almost everything we do turns out to be significant. This is a thing we see in genetics all the time, genomics all the time. And so what I can say, so this is the covariance of S. Average covariance of S over all the sites, 1 interval apart, 2 intervals apart, 3, 4, and 5. And you can see some years they're negative. So it means S in 1 year is positive, the next year it goes down. This doesn't mean there's a sign change, I want to emphasize. It just means they could all be positive, but if you're big here, you're not so big there, and so on. So you get this, I don't know, this is probably serendipity due to weather changes or something. We don't really know what's driving these. What I do want to emphasize, though, these are quite small numbers. So relative to the temporal variance, the covariances are on the order of 1/10 to 1/100th. So there isn't really very much temporal variance in selection going on in these populations. And then I'll mention these islands of strong selection, I guess, right now. There's some regions of the chromosomes that are under stronger selection than others. And so what we've done, now we've got these 1.5 million sites, we've got measures of S up and down all the 12 chromosomes, there they are. And we then break them into windows of about, I think it's 100 meaningful SNPs, and then get an average measure of the intensity in that window, and then do it in the next window, and the next window, and the next window, and so on. And then we're looking for windows with significant selection. These are sort of like what you call Manhattan plots, but kind of different analyses here. The point is that these are each chromosomes. These are the relative lengths of all the chromosomes. And the blue regions, those are the centromeres.
You can call them Sedona plots.
Pardon?
Sedona plots.
Sedona plots.
Red.
No one's going to know what we're talking about. I live in Sedona, part of the time.
Red mountains.
Oh, yeah, I'll show you later. Yeah, we have these beautiful red plateaus. They're mesas. So, good point. I even made them the right color. These are the critical cutoffs, the red lines there. And those are based on randomizations of data and so. But what we're trying to do is now we have significant levels for all these windows, and there'll be a lot of adjacent windows that are significant. And so we just grow the width of the window until we can't grow them anymore. And that's what you see with these bars. So, and we're calling these islands of strong selection. There's actually more of these than you can see. A lot of these are subdivided, but you can't see them on these graphs. But each of these yellow goldish regions are these islands of strong selection. So that's where there's particularly strong selection going on. And you can see the black dots way above the points there. And so a couple points can be made here. There's 122 of these ISSs, and they cover about 10% of the genome. That doesn't mean that we're saying there's no selection elsewhere in the genome. These are just the most pronounced regions of selection. It's the same as when we do Manhattan plots and so on. We don't exclude everything else, but these are the strong outliers. Yeah. Are there centromeric regions?
Yes. Some of those centromeric regions, for some chromosomes, they have a lot of these islands of strong selections, but
Yeah.
some of them, like chromosome 9, for example, or 4, they are depleted. Do you know?
I don't have an explanation for that at this point. I mean, this is a really pronounced one, right? And there could be just some key genes in there. There's a lot of genes in these centromeres, I should say.
And may I ask if recombination is suppressed in this?
Recombination is suppressed. And in fact, I was just going to mention something about the centromeres right now. So overall, these islands are about 50% enriched in centromeres per area. So they are more common in centromeres. They're longer. The islands are about 3 times longer in centromeres. So you kind of expect this. And these centromeres are not gene depauperate. There's a lot of genes in there. And there is recombination going on there, but it is suppressed. So those are the patterns we see. And then this is just, again, the relationship to linkage. So that's sort of the big picture of what's going on at the single site temporal level of selection. And this is just a natural population. It was not picked because we thought there was a specific kind of selection going on out there. We picked it because it's undisturbed and probably will remain undisturbed for the next several decades. And we just wanted to try and understand what the basic background fluctuations are in a normal genome. You can look in those islands and then say, this is probably partly related to your question, are there anything specific going on in these islands? And it turns out 42% of the genes in these islands are young duplicate genes. Daphne have most invertebrates have a lot of duplicate genes, but this is a lot higher than the 30% in the non-ISS region. About half of these genes are Daphnia specific. We know they're expressed, but we don't find them so far outside of Daphnia. There's a lot of other arthropod genomes sequenced. Now, this is what gets interesting. The ISS genes, they have lower levels of silent site diversity. than other genes. And as a consequence, the ratio pi n pi s is about 25% higher. You know, people tend to think of a high pi n pi s either meaning relaxed selection or maybe some kind of balancing selection on the non-synonymous sites. Maybe, but I'll go through another possible explanation in a second. The ISS genes have similar levels of silent site divergence as everything else. We expect that to be the case if the silent sites are really neutral. Doesn't matter how much recombination is going on. If you're neutral, that's the rate of evolution. But they have elevated rates of change at replacement sites. The main thing that this implies, all these measures, pi and DNDS, these are our long-term measures, right, of genes. They're factors of past history. And yet the study I've been telling you, that was only a 10-year study. But this suggests that the genes in these islands versus outside of the islands are reflecting something that's been going on for longer periods of time. So one theory slide here. It's taken me a while to get my head around what's exactly happening here. And we don't have all the theory worked out yet. And there's a lot of ways to think about this. This fluctuating selection could be on quantitative traits, for example. Probably is to some extent. And we shouldn't be thinking about just a single locus thing one at a time. And there's also the problem of linkage, right? You're a victim of everybody you're in LD with at any one point in time, and LD is going to be changing through time. That might be causing fluctuating selection. But I do want to point out some relationship to preceding theory and those estimates of pi n pi s I just mentioned. So quite a long time ago, I guess it's 50 years ago, Sam Carlin wrote this paper working out the site frequency spectrum for alleles experiencing fluctuating selection. And this is turns out to be very similar to site frequency spectrum for an allele not under selection. So in fact, if the denominator here were 1, that would be the site frequency spectrum for a neutral allele. And this theta is our friend for NEU. And this quantity here, what is that, a gamma? Gamma, we'll call it gamma. That's for any type sigma squared s. So that's the scaled measure of fluctuating selection. And you can see if this number here is a lot less than 1, fluctuating selection doesn't do anything. The site frequency spectrum should be just like neutrality. But remember, our average value of this is 0.01, and this is about 600,000. So we have big values of this. So this is going to influence the site frequency spectrum. Now, if you take the site frequency spectrum and multiply by 2x1 minus x, heterozygosity, and integrate, you can get an estimate of the expected heterozygosity at a site. One other problem, though, with this formula, and a lot of the formulae we use like this, there's an NE in here, and you're just given this NE. But it's a little unclear to me if fluctuating selection itself generates NE. And so it's really nice to have this math worked out, but we decided to do computer simulations just to check it all out with different values of the mean and the variance. And the main thing, I just want to point out 2 things. What the results show is that this is the relative neutral variation relative to complete without selection. And you can see that under this model, you get reduced levels of silent site variation. This is at the silent sites, a reduction in pi s. It's exactly what we see. And you get elevated levels of pi n pi s. That's exactly what we see. And I was trying to work out the math here and really getting a headache. And one day I was talking to Archana Devi, who was in my lab at the time, and I'm thinking, somebody must have figured this out using the coalescent. And I just went to my computer and plugged in coalescent fluctuating selection. I thought I had read all the papers, and up came this paper by Jesse Taylor. who was Bruce's only graduate student ever, I think, and I've learned that he was a postdoc with Nick. And he's my colleague in the math department at ASU. And he had worked out all the math in a theoretical population biology paper. And he showed, using the coalescent, that this is the behavior you expect. With fluctuating selection, it gives you a shallower coalescent. And then that reduces the silent site variation. But it reconfigures the positions of the two alleles, the big A and the little a. They tend to be on opposite sides of the tree, so that gives you elevated pi n. Pi n is still depressed relative to what we do out selection, but not as depressed as pi s. And so I think the theory is quite consistent with the results we're seeing. So there's that. And the theory also predicts an elevated rate of fixation when you have fluctuating selection, elevated rate of fixation of the selected site, not of the non-selected site, because that's truly neutral, but of the selected site. And that's also exactly what we see. I think it's telling me, okay, I'm going to stop with this one slide. There's the other stuff that I never finished up that I was going to talk about, but I think people are probably getting tired. Be careful what you say or take a vote. Let me point this out, though, some biology. Remember, we have this 10 metapopulation study, and then we have 10 years of study in one population. And so for every gene, we've got an estimate of pi n, pi s, dn, ds. And from that, some of you are familiar with this thing called the neutrality index. That's the ratio of pi n, pi s to dn, ds. And the idea is if the neutrality index is, if pi n, pi s is low relative to this, it suggests positive selection, causing divergence over time. So low values here would mean positive selection. And high values mean stabilizing selection. If you can't diverge, that's going to cause this ratio to go way up. This is a.
If you have such thing as very deleterious, that changes the pi n ratio.
Oh yeah, that will influence this a bit, but you need something also to pull this up. So the main point is this. We have a lot of data, 1,000 genomes from 2 different studies, same species, populations are in the same region. And one question that could be asked was, do you get consistent results with these different data sets? How noisy are all these estimates on a gene by gene basis? And here's the bivariate plot from the 2 approaches. And so these are genes that have a neutrality index that are is not significant from the neutral value of 1 in both populations. These are significant in one, not the other. And then these are significant in 2. So those are genes that are really giving us strong indications that they're in this species anyhow, they're in strong positive, experiencing strong positive selection. And quite remarkably, there's sort of a story emerging here. These genes are very, very highly enriched with ribosomal protein coding genes, both for the cytosolic ribosomes and the mitochondrial ribosomes. And this is why we're now trying to get the structure of the ribosomes in these bugs to figure out what's going on. So even the mitochondrial ribosomes, the proteins around them are all encoded in the nuclear genome. They're also very enriched in other nuclear encoded mitochondrial genes, like the ones in the electron transport chain and ATP synthase. And they're highly enriched in components of the nervous system, in particular opsins. These are the proteins the bugs use to sense light. They're very light sensitive for vertical migration, olfactory receptors, and other kinds of environmental sensing. So it's at least starting to tell us about sort of some of the biology that might be interesting to pursue. I'm going to skip over this and just give you a few closing remarks. And one thing, if anybody's looking for a postdoc, even faculty, I got a weird note from my program officer last night. I've been waiting on a grant for like 14 months. My program officer forgot to tell me I had the award. It's been sitting there, and now I'm in trouble because I haven't spent a dime on this grant. You know, it's good news, bad news. Anyhow, if you're interested in any of this or any other theory that I talked about, please talk to me. And then I'm gonna skip over this. Sorry, I know Juliet really wants to hear this. Good timing. Just a couple closing slides here, if I can get there. 3 closing slides. Okay, these are the people who have contributed to this work. Really great people. Mike Frender and Andy Zeloff, my colleagues, left back in Indiana. They're going to be the victims of keeping this study going forever, where we do the sequencing and help with the analyses. And then these are our funding agencies. This is what I used to do. I got introduced in Daphne a long time ago when I did limnology. So one of those people up there is me. And we used to, we were very interested in lake management. We were trying to figure out how to restore lakes by adding zooplankton, Daphne, to graze down all the algae without trying to control phosphorus and so on. That's my advisor. His nickname was Mad Dog Shapiro. And we had an amphibious car. This was great fun. We were able to just drive out into lakes in this car. We would drive down the highway and with propellers on the back, and we'd go into the middle of the lake, and some little old lady would come out and call the police, and police would come out and say, "Get that car out of the lake." And my advisor would always, who couldn't swim, by the way, that's why we had the car, and we'd always start screaming them with a bullhorn, "We are limnologists." So you don't get the joke if you don't know what a limnologist is. We did a lot of splitting lakes in half and doing manipulations. My advisor was interested in effects of CO2 on lakes, pH. What better way to get CO2 into a lake but dump a tractor trailer of dry ice into the lake? That's the result of that. Shut down the state highway, and quite a few cars from the state police showed up on that one. That's my former advisor. That's the first undergraduate in my lab, Bob Sterner, who eventually became his boss in the department I got my PhD with. That was interesting. And then finally, I'll just close with this. There's fly people in the room, and all you fly people will know who Morgan was. And there was this place where it all started in Columbia in the fly lab. It's not there anymore. But this may be an urban legend, but it's in this great book called Lords of the Fly. If you work on flies and you haven't read this book, you need to read this book. Students who asked Morgan to give them a practical problem were likely to be put off with a practical joke. Edgar Altenberg, I'm not sure if he's related to Lee, suffered from such a joke. Asked for a dissertation topic, Morgan took him across the hall into the aquarium room, dipped his finger into a stagnant tank, scrutinized the green liquid for a moment, and then suggested Altenberg might work on Daphnia. To Altenberg, who desperately wanted to be a Drosophilist, it seemed like a calculated rebuff, and he decamped for the Department of Botany. So I guess that tells you what Morgan thought about Daphnia. He was exploring all kinds of organisms, but Daphnia wasn't his first choice, unfortunately. That's it, thanks.
You need to answer your question.
Oh yeah, so the question, I'll turn my back. You may think that that long-term experiment is just, why are you doing this? Or this is going to be an amazing data set in 20 or 30 years for people to explore just basic patterns of allele frequency change. We aren't doing any ecology. And I feel kind of bad about that, but the more I think about it, we could have studied 50 different ecological factors. At this point, I would have no idea what was driving the allele frequency change, I don't think. But maybe if we did all those 50 every year for 40 or 50 years, this would really emerge as a useful thing. And we aren't phenotyping the bugs either at this point. I mean, there's just so much we can do. Anyhow, Bruce will take a tally, and I'm just interested if you think it's worth going. we're not keeping the resting eggs. The limit here is if we make resting eggs of those clones we bring in, we would be selfing the line. They won't be the same genotype. We are keeping the clones as long as we can, but we can't keep 100 clones a year for 20 years. That's 2,000 clones for, it's just impractical. So I guess the question is, I'll leave the room. I just be interested in a tally or suggestions, what you think might be done here at relatively low cost, because we're just sort of doing this on the back burner relative to the other things we're actually funded for.
So I would, I would point out that the Yeaman involved experiment kind of arose by a similar thing. Once you kind of got it going, you thought, well, this is working, you might as well keep it up because no one else is doing it.
Isn't there a thing called, what is it called, the Concord fallacy? Is that relevant here? I don't know. We've kept it going for 2 more years, and we will probably try and keep it going for a bit more.
So maybe this is a silly question, but can you, because you followed every single year, and can you also predict from 1 year to 10 years beyond? Like, could you change the frequency of sampling if needed and sample every 10 years instead of every year?
How old do you think I am?
Every 20 years, you sample.
Mike Frender's maybe 10 years younger than me. But maybe that's, the problem is if you do that, then we're trying to do, intervals of years for S's. And so we would lose that. It might be adequate for looking at the long-term trajectory. But
The problem here is the sampling, because the genomics is tricky.
yeah, the genomics is trivial, the sampling, and I think we could design field work, but the problem is I live 3,000 miles away from that site now. So that makes it a bit difficult. And there are no lakes and ponds in Arizona. No natural lakes and ponds. Okay.
What answer would you like to get?
Pardon?
What answer would you like to get?
Well, I would like to be able to get these measures more refined. Those variances are still pretty noisy. And then I don't know, I would like to see if those covariances that we measured, they're probably pretty serendipitous, just, changes in weather patterns in, like a 9-year period. But what if you looked at, adjacent periods over, a span of 20 or 30 years? There might be some more general patterns. I mean, I think if you're an ecologist, you would tend to think that there'd be a dissipation of the covariance of selection as the distance increased, but that's not what we're seeing so far.
So would it be an idea to basically standardize the molecular methods that we use for you still have DNA or something like this? You basically use the same sequencing platforms and all this to see how you can minimize the noise. You cannot account for standard binomial sampling that comes from different sequencing technologies. I don't know. I'm very skeptical of the noise. Do you think this would be maybe as informative as adding more years?
Well, I think our estimates of loop frequency are as good as they get. We're using a maximum likelihood technique that factors out the error. But we can't go back on the technology. We're not going to go back. I guess we never used 454 sequencing, but we're never going to go back to the original Illumina sequencer.
No, but you can use the old sampling for the new technology.
Oh.
DNA left.
Yeah, I don't know if we still have the DNA. That's a good idea, though. If we do, and even if we don't, that would be a good idea for the future to make enough DNA for future analyses. We might still have it.
I also have a question regarding the deleterious variants. When people don't know how the frequency changes, you use the GERB scores or SNPF or sort of inference of how deleterious a mutation might be based on the effect on the protein. Do you find an association between the S you observed and the effect of the variants on the proteins, the severity of their effect?
Oh, we haven't looked in incredible detail, but we've looked at the average S for silent sites and replacement sites and intron sites and so on. They're pretty similar. They're within just a few % of each other. And I think that's not too surprising for a study this short term, and given you've got this linkage window, there probably are real meaningful differences. In fact, those differences, a few % are significant, but maybe not as pronounced as you'd think from, you know, classic ways we think about silent site and replacement sites. Okay, well, I think everybody's probably pretty wiped out at this point, so, and you need to get coffee into people before, I think Nick is next. Okay.